Imagine a world where a simple scientific pursuit could potentially unleash an existential threat to all life on Earth. It's a chilling thought, isn't it? This is the story of a scientist's journey, a journey that began with a simple idea and ended with a global conversation about the boundaries of scientific exploration. Meet Kate Adamala, a synthetic biologist with a curious mind and a passion for pushing the boundaries of what we know. In 2019, she and her team embarked on a quest to create something extraordinary, a mirror cell. A cell unlike any other, where all its components would be the reverse of what we find in nature. A cell that could potentially shed light on the origins of life itself. But as the team delved deeper into their research, doubts began to creep in. What if this mirror cell, this artificial creation, were to escape the confines of a lab? Could it spread unchecked, like a rogue element, posing grave risks to human health and the delicate balance of our planet's ecosystems? The concept of chirality, or handedness, in biomolecules is a fascinating yet complex idea. Just like our hands, many major biomolecules are either right or left-handed. This property, first discovered by Louis Pasteur, is what makes interactions between molecules so specific and unique. DNA, RNA, and proteins all have their unique chiralities, and any disruption to this delicate balance could have catastrophic consequences. The idea of creating a mirror cell where all these molecules are reversed was an exciting prospect. It promised to unlock new therapeutic possibilities and tackle some of the most pressing medical challenges we face. But with great power comes great responsibility, and as the team realized, the potential risks were immense. It wasn't an aha moment, Adamala recalls. It was a slow realization over months. We thought we could answer the questions, but then we realized we couldn't. And here's where it gets controversial. As the team consulted with experts in biosafety, immunology, and ecology, a worrying picture began to emerge. These mirror cells, they realized, could be invisible to our immune system. Like a stealth invader, they could replicate and spread without our bodies. Natural defenses even noticing. I used to think the immune system would find a way, Adamala admits. But I didn't know how chiral our defenses were. Fast forward to 2024, and the concerns of Adamala and her colleagues had grown into a full-blown working group of 38 scientists. Together, they published a groundbreaking article in Science, titled Confronting Risks of Mirror Life. Their report concluded that mirror cells could become a reality within the next few decades, and outlined the devastating consequences if these artificial life forms were to escape into the wild. Since then, a non-profit organization, the Mirror Biology Dialogues Fund, has taken up the mantle, sponsoring meetings to develop strategies to mitigate this potential threat. The question on everyone's mind: where do we draw the line? Should we even be exploring this field of mirror biology at all? At a 2-day conference in Manchester, UK, experts gathered to discuss these very questions. David Relman, a professor of microbiology and immunology at Stanford University, attended the meeting and shared his concerns. There is a real possibility that we could create something that could grow uncontrollably, spread across the planet, and displace or kill many forms of life, including us. Relman, like Adamala, was part of the early working group. He remembers keeping their initial conversations under wraps, not wanting to sound alarmist or like conspiracy theorists. But as their research progressed, the potential risks became harder to ignore. The more we looked, the more certain we became, Adamala says. There is no safe way to make a mirror cell. For Relman, mirror life represents the first plausible existential risk he's encountered in his long career. He's investigated deadly anthrax letters and the mysterious Havana syndrome. But mirror life is different. It doesn't exist yet, and that, he believes, gives us a unique opportunity. There's a chance we don't have to go down this path unless we choose to, he says. But not everyone agrees. Other experts argue that we should tread carefully, not making knee-jerk decisions that could stifle scientific progress. They distinguish between the research on individual mirror molecules, which hold therapeutic promise, and the more controversial pursuit of creating mirror cells or organisms. Michael Kay, a professor of biochemistry at the University of Utah, is focused on developing drugs based on mirror image molecules. He's not in favor of blanket regulations that could block an entire area of study. Mirror molecules are inert chemicals with tremendous benefits, he explains. They're here, and they're going to be a big class of drugs in the next few years. Kay acknowledges the unknown risks of mirror cells and organisms, but he believes the ongoing risk assessments are important. Any effort that buys us time to better understand and consider the risks is beneficial. This is not an imminent threat. Many synthetic biologists, including Adamala, are focused on creating a synthetic cell with natural chirality from scratch. The aim is to mimic biological processes and solve problems in medicine, industry, and basic research. A synthetic cell, Adamala explains, would be like an operating system for life, allowing us to engineer biology with unprecedented precision. We will build models of cellular processes to understand how healthy cells work and how diseases start. She says, according to John Glass, the professor at the J. Craig Venter Institute, we might soon reach the milestone of creating a synthetic non-mirror cell. And if we can create a normal cell with natural chirality, he believes we could theoretically create a mirror image cell using the same methods. Glass's initial conversations with Relman about the potential risks left him shaken. It made me wonder if my work could one day enable the mirror bacteria-based Armageddon we fear. Most experts agree that creating a synthetic cell with natural chirality is safe. If such a cell were to enter an environment, it would be subject to the normal controls of any ecosystem, making it easy prey for natural predators like viruses. For Glass, a clear red line is refraining from making a mirror ribosome, a biological machine that makes proteins. Kay, however, has a different perspective. His lab is focused on improving the synthesis of mirror proteins, and he's interested in exploring the idea of a mirror image ribosome. The idea is to make these products with higher quality and longer proteins, which might be compatible with pharmaceutical use, he explains. But there is concern whether having this tool would make the development of mirror life inevitable or too easy. Adamala and her colleagues chose not to renew their research grant, ending their work on mirror cells. Instead, she's focusing on discussions around regulating mirror life research. To her knowledge, no scientists are actively pursuing the creation of a mirror cell. Pretty much everyone agrees we shouldn't make a living mirror cell. That's the baseline. But below that, people have different ideas about where to stop the research, Adamala says. The scientific community can't agree on the red lines. Relman hopes their proactive efforts will not only protect the planet, but also help rebuild trust between scientists and the public. We scientists think about whether we should, not whether we could. As the discussions continue, the future of mirror life research hangs in the balance. Will we choose to explore this uncharted territory, or will we err on the side of caution? The decision, it seems, is ours to make.